All right, sorry about that. The video cut off, but I'm going to pick it back up at the same slide. So we have complete antigens and haptens. That's what we're talking about. Remember, we had complete or incomplete antigens, and then incomplete antigens are also known as haptens. Now, in order for an antigen to be a complete antigen, it has to have two important functional properties, and they are immunogenicity and reactivity. With immunogenicity, we're talking about having the ability, so the antigen has the ability to stimulate the proliferation of specific lymphocytes. Reactivity just means that it's going to be able to react with activated lymphocytes and antibodies that are released by immunogenic reactions. We have some examples of complete antigens. They're going to be foreign proteins, polysaccharides, lipids, a number of other things. Then we have incomplete antigens, also called haptens. Okay, so these themselves, so haptens, are too small to be immunogenic themselves. And so examples of this are going to be small peptides, nucleotides, some hormones. Now, what can happen is these haptens actually attach to proteins in the body. And then that combination of the hapten and the protein at that point is seen as foreign. And once that's seen as foreign, the immune system mounts an attack that ends up being harmful to the person because it's going to go after the self protein as well as the hapten. Examples of this are going to be poison ivy, animal dander, detergents, cosmetics. Okay, so remember we were talking about the fact that there were a couple of different types of antigens, and then I mentioned that we were going to look at antigenic determinants. And these are the parts of the antigen that antibodies or lymphocyte receptors bind to. That's it. It's just literally the part that the antibody or lymphocyte receptor binds to. So here, these would be the antigenic determinants. These little kind of areas here and here and here, you see them binding to the antibodies, right? And so the part that's binding to the antibody is going to be that antigenic determinant. And we also have self-antigens. So all cells are going to be covered with a variety of proteins. And typically, for the most part, they're not antigenic to self, but they can be antigenic to others in transfusions or grafts. So we have antigens, say, like on our blood cells. And if I have type AB blood and I donate to somebody who has type O blood, then there can be some reactions that occur from that. All right. And now we move to lymphocytes and antigen presenting cells. So the adaptive immune system is going to involve three really important cell types. Two of them are lymphocytes. So we have B lymphocytes, which are also known as B cells, playing a role in humoral immunity. And then we have T lymphocytes, also known as T cells, which are going to play a role in cellular immunity. In addition to the two types of lymphocytes, we have antigen presenting cells. These actually do exactly what it sounds like they do. They are going to present an antigen. They do not respond to specific antigens. They play auxiliary roles in immunity. And auxiliary means providing additional help or support. So basically, they play these like real supportive roles in immunity. All right. Now we move on to talking about lymphocytes. And so we're going to talk about lymphocyte development, maturation, and activation. And there are going to be 5 general steps. And these are shared. So they're actually they're common development steps in the life cycles of both T and B lymphocytes. So the first step is going to be origin, and this is simply that both B and T cells originate in red bone marrow. So, cursor. Okay, so we have the origins in red bone marrow. Maturation is second. We have the lymphocytes are educated in a couple 2-3 day process, and they're going to mature in primary lymphoid organs. Remember, primary lymphoid organs are the bone marrow and the thymus, and B cells are going to mature in the bone marrow, B for bone, and the T cells are going to mature in the thymus, T for thymus. So maturation, we're going to have the B cells maturing in bone marrow, the T cells maturing in the thymus. The next thing we're going to have happen is the secondary lymphoid organs are going to be seeded, and circulation of lymphocytes. Basically, what we're going to have is these immunocompetent B and T cells, and they're going to be called naive. And naive immunocompetent B and T cells are cells that have not yet been exposed to an antigen. So these naive cells are going to be exported or sent out from primary lymphoid organs, again, B cells, bone marrow, T cells, thymus, to colonize secondary lymphoid organs, such as the spleen, lymph nodes. What this is going to do is this is going to increase chance of encounter with an antigen. Remember, a lot of our kind of garbage gets swept up in the lymphatic system, and so it's good to have the lymphocytes like in the lymph nodes to kind of help catch pathogens that can end up in in our lymph. So we have seeding of secondary lymphoid organs. So it's going to be going to the bone marrow, lymph node, spleen. Then we have antigen encounter and activation. So a naive lymphocyte will encounter an antigen, and it's going to actually trigger the lymphocyte to continue to develop. The lymphocyte will actually differentiate into an active cell whenever it binds to a specific antigen. And then just basically, this is the next line is just saying that if the correct signals are present, the lymphocyte will complete the differentiation into an active cell. So we have our naive cells were kind of hanging out in the lymph node. They encounter an antigen, and once that lymphocytes antigen receptors bind to the antigen, the lymphocytes activated, and it can, complete that differentiation into an active cell. Finally, we have proliferation and differentiation. Once the lymphocytes have been selected and activated, they proliferate. They form basically just clone armies. I mean, I'm guessing many people know something about clone armies with Star Wars, I think it is. Anyway, so what they're going to do is they're going to form these armies, exact copies of themselves, and they're going to be clones. Most clones become effector cells. They're going to fight the infection. Some are going to become memory cells, and they're going to be once they're memory cells, I think I mentioned this earlier, they have the ability to respond to that same antigen more quickly whenever it's encountered a second and subsequent times. And then we have B and T memory cells and effector T cells circulate continuously. And this is just the last part with the proliferation and differentiation down in here. And then the memory cells and effector T cells will circulate around. Memory cells can last years. All right. So remember, we talked about the fact that, or I guess I should say, I talked about the fact that there are three important cell types with adaptive immunity. There were the two types of lymphocytes, the T cells and B cells, and then the antigen presenting cells. And we're going to talk about antigen presenting cells now. What they're going to do is they're going to engulf an antigen and present a fragment of that antigen to T cells for recognition. It does exactly what it sounds like it does. It just presents an antigen. The major types are dendritic cells. They're going to be the most important, macrophages, and B cells. Dendritic cells are found in connective tissues and in the epidermis. And what they do is they are just like the guards of boundary tissues. They phagocytize pathogens, they eat pathogens that get into or enter the tissues. And once that happens, they enter the lymphatic system and presents the antigens to T cells in lymph nodes. They are the most effective antigen presenter known, and they are the key link between the innate and adaptive immune systems. This is a dendritic cell. The next type of antigen presenting cell, or APC, is a macrophage. And these are widely distributed in connective tissues and lymphoid organs. And they also present antigens to T cells. But the cool thing about macrophages is that whenever they present the antigen to T cells, not only does it activate the T cells, but it actually kind of like supercharges the macrophage. So once it supercharges that macrophage, that activated or supercharged macrophage becomes a voracious phagocytic killer. It's also trigger powerful inflammatory responses and recruit additional defenses. And then finally, we have the B lymphocytes. They actually do not activate naive T cells. What they do is they present antigens to helper T cells to assist their own activation. This is just a table kind of going over the B and T lymphocytes, and we're going to get into more detail in all this in subsequent videos and class time. But that's all I have for this video.